Good day everyone, we are the Group 1 and we are here to present our lab report in regards with the Experiment 1 qualitative test for proteins.
Here are the group members. Chinchantic Mark Ian, Liman Paula May, Rias Aris Joseph, Gayosa Catherine Michael, Lagudas Hilda Luis, Lontok Russell, and Morales Stephen.
Here is the content of our lab report. Includes introduction, methodology, procedure, results and discussion, conclusion, recommendations, and references. Proteins are long chains of amino acids held together by peptide bonds. Proteins make up half of a cell's dry mass. These four elements, carbon, hydrogen, oxygen, and nitrogen, are present in all proteins. 20 common amino acids are known to exist, and each one has a unique side chain. The structure of a protein is determined by its amino acid sequence, and the structure determines its function. What is the importance of qualitative tests for proteins? The different R groups or the side chains and their chemical interactions with different reagents are used to identify and characterize the presence of individual amino acids in a sample in order to assess proteins. However, because proteins contain a variety of amino acids, one test will not be sufficient to detect amino acids. Before making any assumptions regarding the composition of a protein, it is advisable to do several tests. Qualitative protein test is necessary to identify proteins and understand their structure in biological materials. It is beneficial for food safety, disease diagnosis, and enzyme activity research. Scientists and medical professionals can use specific proteins or amino acids to identify health, track enzyme activity, and research cellular processes. This study ensures the pharmaceuticals and food production.
Methodology. We use the various methods in order to determine whether the samples contains proteins. The following tests performed in this experiment are ninhydrin, urette, Milon's, Hopkins Cole, Xanthoproteic, and False test. These tests, we use this in order to determine the presence of each samples of each of their proteins.
Here are the materials and reagents used for this experiment. Please refer to the table provided. For the procedure, in this experiment, you need to identify different amino acids and evaluate contents of protein and unknown samples by applying qualitative tests. So the first procedure is preparation of samples. Get around 15 ml of each samples, fresh egg white, fresh milk, alanine unknown to in your vials. For qualitative test, in test A, ninhydrin test, get 2 ml of each samples in a clean, dry test tubes. Add 10 drops of 2% ninhydrin solution. Heat in a hot water bath and observe for any change in color. Letter B, urette test. Get 2 ml of each samples in a clean, dry test tubes. Add 1 ml of urette reagent. Shake well and take note of any change in color. For C, the Hopkins-Coles test, get 2 ml of each samples in a clean, dry test tube. Add 1 ml of glycosylic acid and carefully run 1 ml of concentrated down the side of the test tube. Make sure that two liquids do not mix. They should separate into two different layers. Take note of the color that forms between the two layers. For D, millions test, get 2 ml of each samples in a clean, dry test tube. Add 10 drops of millions reagent. Shake well and heat in a hot water bath. Observe for any change in color. For E, Xanthoproteic test, get 2 ml of each samples in a clean, dry test tubes. Carefully add 1 ml of concentrated. Take note of the mixture's color. Place on a hot water bath for about 20 to 30 seconds until a yellow precipitate forms. If the test tube feels hot, allow it to cool down by running the tube under tap water. Carefully add 2 ml of 40% NaOH solution. Note the color of the mixture. For F, false test, get 2 ml of each samples in the clean dry test tubes. Add 1 ml of 20% NaOH solution and 10 drops of 10% Pb2 solution. Heat the tube in a hot water bath and observe for any color change.
Comparison of actual versus expected results. For fresh egg white, we expected positive results because of the above above. The Burette, Hopkins Co, Santoprotein, and Foles test are supposed to show proteins, tryptophan, aromatic amino acids, and sulfur-containing amino acids, respectively. We don't expect melons to be positive since egg whites don't have much tyrosine. In the actual results of our experiment, everything went as expected. The egg white tested positive for ninhydrin, showing free amino acids, burette, confirming proteins, Hopkins Co showing tryptophan, Santoproteic indicating aromatic amino acids, and false showing sulfur-containing amino acids. The Milan's test was negative, which makes sense because there's not enough tyrosine in egg whites. For fresh milk, we expected it to test positive in most of the protein tests because of casein and its free amino acids. Just like the egg white, we didn't think it would be positive for melons since that test looks for tyrosine and milk doesn't have a lot of that. The actual results matched what we thought. Milk showed positive for everything except melon's test. This confirmed that milk has proteins, free amino acids, and tryptophan, with a negative melon's test means there's not much tyrosine in it. For alanine, we thought it would show positive for ninhydrin and burette tests because it's a simple amino acids, but negative for melons and Hopkins Co since it doesn't have phenolic or tryptophan groups. The actual results were pretty much what we expected. Alanine tested positive for ninhydrin showing free amino acids and burette indicating peptide bonds. It was negative for melons and Hopkins Co, which makes sense since alanine doesn't have the stuff those tests look for. For the unknown sample, we weren't sure what it was, but we thought it might have proteins or amino acids. The actual results showed it tested positive for burette and melon's test, meaning it has proteins and probably tyrosine. But it was negative for ninhydrin, Atkins Cole, and false, which means it doesn't have free amino acids, tryptophan, or sulfur-containing amino acids. So we think the unknown sample is something protein-rich like casein, but without sulfur or free amino acids.
In conclusion, the qualitative test effectively demonstrated the presence of proteins and specific amino acids in each sample. Fresh egg white and fresh milk showed positive reactions in multiple tests, including burette for peptide bonds, ninhydrin for free amino groups, and xanthoproteic for aromatic groups, confirming their richness in protein. They also tested positive for Hopkins skull, indicating indole groups like tryptophan, and false showing sulfur-containing groups like cysteine. Alanine's results confirmed its expected structure by testing positive for biuret and ninhydrin, but it did not show the presence of indole, phenolic, or sulfur groups. The unknown sample tested positive for burette, melons, and xanthoproteic, indicating it has tyrosine-based proteins, but lacks other groups like free amino groups and sulfur-containing amino acids.
As for the recommendations, first, let me first the lab recommendations. The Burette test ensures students must understand the molecular basis of the violet complex, as long as the non-hydrin test ensures safety protocols are followed. And for the Milon's test, ensures safety protocols are followed due to the use of concentrated acid. And Santropotoic test, it is highly specific. That's why it requires careful handling of the reagents. As as for the Hopkins Cole test, students should be careful when handling sulfuric acid. In the last but not the least, the false test ensures students handle the reagents carefully and understand that this test is specific for sulfur-containing amino acids. As for the general recommendations, we also add the sample preparations. safety protocols, comparison and controls, and visual recordings.
Our reference, we have solely based on the datasheet and post lab of Group 1.
And that concludes our lab report. Thank you so much for listening.